Hello, my name is Ekaterina. Let me present a short video tutorial on our new version of the query page of CYTOCON DB. CYTOCON Database is developed by InSysBio. It is the database that contains baseline concentrations of cells and molecules in both healthy and suffering from various pathologies human organisms. Let's have a look at our query page. Here you can see several fields. Let's fulfill them to obtain the information from the database. Within Select Species Type field, I select the type of species, for example, cell, and in Species field, I choose B cells. Here is the field Tissue Type, I choose blood. I can choose 2 or more tissues in the field. And here I select the disease from the list. Healthy control, for example. Here is the button Cell Markers. I can choose a number of markers to find the information we need. For example, I'll choose this. You can also choose additional disease attributes and additional patient group attributes here. And then we have to press Query button. After that, we obtain the table results with a number of columns containing species name, markers, species value in unified units, which are kilo cells in liter in this case. CYTOCON DB team reconsiders all the published results so that they can be easily compared in the database. We can also find here dispersions, mean and max values, original species values in original units from the articles, and the information on units, tissue types, diseases, the types of average and dispersion, and the links from the article are here. There are also columns containing patient group attributes and disease attributes. Here they are. We can display them all, or you can find the buttons here, hide patient group attributes and hide disease attributes, so we can hide them or open these columns. Next, we can export all this information to CSV file, pressing this Export to CSV button. Then we obtain the file that can be opened in Excel and further analyzed. Here is also Graph Result button. By pressing it, we obtain the figure that shows means and dispersions in unified units. Once we have made a query, we can save it for the future in Query Manager. It is here. We have to give a name to our query which was made. I'll type here, for example, B cells, the name of species, and the name of tissue, and the name of disease. And we press Save Query button. After that, this new query appears in the list of the earlier saved queries. It can be found here in the list of queries. Here it is. If we don't need to save some queries, we can delete them via Delete Selected Query button, like this. Then we can return to the query parameters to make a new query. For example, we can choose earlier saved query. You see that the fields are automatically fulfilled after that, and we can press Query button and obtain new table results with new information, and graph result is here. Then we can return to query parameters on the query page. So I have shown you the possibilities of our query page of CYTOCON database. Thank you for your attention and goodbye.